Hello students, welcome back to the biology class. So in the last class we started the second type of dark reaction possible, that is the C4 pathway. We learned that it was explained by Hatch and Slake, that's why it is also called as Hatch and Slake cycle or Hatch and Slake pathway. It was explained by them in 1966, and because there is 2 times carbon dioxide fixed in these plants, that's why this cycle is also called as double carbon dioxide fixation cycle. The first carbon dioxide fixation occurs in those cells which are closer to the stomata, and those cells are mesophyll cells. And the second carbon dioxide fixation occurs in the bundle sheath cells. Yes, these cells do not carry chlorophyll in a normal plant, or normal plant means C3 plants, but in the C4 plants there is an adaptation. Even the bundle sheath cells carry chloroplast, and that's why there is 2 times carbon dioxide fixation. C4 pathway involves 2 types of cells, bundle sheath as well as mesophyll cell. But remember, C3 pathway also occurs in these plants. So these C4 plants will have 2 types of pathways. That's why there is 2 extra ATP required to fix the carbon dioxide. But the good thing is that they are more efficient in performing their activities. So there are 18 angiospermic families, 3 monocot families and 15 dicot families. That means total 1500 species of plants show C4 pathway. Some of the examples we learned in the last class. And we learned that in the tropical, subtropical, high salinity, dry condition and high temperatures are the conditions which favor such type of plants. So these plants have evolved in such type of ecological conditions. And we learned that C4 pathway performing plants are called as C4 plants, and they have a special anatomy called as Kranz anatomy, in which 2 types of photosynthetic cells are there. Then we learned that there is a symplastic continuity between these 2 types of cell. And symplastic continuity means protoplasm of one cell is in link with the other protoplasmic cell, and this is all because of the plasmodesmata or cytoplasmic bridges. We learned that there are 2 types of chloroplasts in these plants, or in these photosynthetic cell, granule and agranule. Grana is absent in the case of bundle sheath cell, and it is present in the case of the mesophyll cell. The chloroplasts are smaller in mesophyll cells and they are larger in the bundle sheath. So the bundle sheath cells do not have oxygen liberation, and they perform only cyclic photophosphorylation, and there is only rubisco present in them. And in the case of the mesophyll cells, they perform C4 cycle and they perform non-cyclic photophosphorylation. So there is oxygen liberation in bundle sheath cell. And then we learned that there is a different enzyme. So they do not have rubisco, so they cannot perform C3 cycle. They have pepcase present in them. The first stable compound formed in C4 pathway is in the first carbon dioxide fixation in these plants is in the mesophyll cells, and then this carbon dioxide which is fixed is transported as a 4 carbon compound. That's why it is called as C4 pathway. And this 4 carbon containing compound shifts through the plasmodesmata and enters into the bundle sheath cell. So we started the biochemical pathway also in the last class, that we understood that there are 2 fixations in these plants. First fixation occurs in the mesophyll cell, and then the acceptor is PEP or phosphoenol pyruvic acid, which is a 3 carbon compound. It picks up the carbon dioxide, a 1 carbon compound, to form 3 + 1 4. So oxaloacetic acid is a 4 carbon containing compound. And this oxaloacetic acid is formed in the presence of pepco or pepcase enzyme. The full form can be phosphoenol pyruvate carboxylase enzyme. Remember, this enzyme has no affinity for oxygen. Remember, rubisco has affinity for oxygen. That's why rubisco is also called as, full form is also like RUBP carboxylase oxygenase. But in this case you simply call it as carboxylase because it does not have the ability or affinity for oxygen gas. Now this oxaloacetic acid undergoes either the reduction or it undergoes amination and then finally change into malic acid, or it changes into aspartic acid. This is amino acid, this is a normal organic acid, and either of them, whatever is formed, will be transported from mesophyll cell to bundle sheath cell through plasmodesmata, and this step is called as transport. Once transport gets over, inside the bundle sheath cell, these acids are going to undergo decarboxylation or release of the carbon dioxide. Once they liberate the carbon dioxide enters into C3 pathway, and the resultant form would be now 4 carbon has lost 1 carbon, so it becomes a 3 carbon compound which is called as pyruvic acid. This pyruvic acid will go back into the mesophyll cell. Now the RUBP undergoes C3 cycle to form PGA. PGA travels back to the mesophyll cell through plasmodesmata, and there it changes into PGAL. Now which will again come back to bundle sheath cell. Now this step is something which is crucial because this PGA first shifts to mesophyll and then turns into aldehyde and now comes back to the bundle sheath cell. So this step is crucial. If the PGA straight away change into PGAL in the bundle sheath cell, this will cause the liberation of oxygen, and then there will be problem. That's why these plants have an adaptation of shifting PGA to mesophyll so that it can change into PGAL, and then it comes back to bundle sheath cell and undergoes changes to turn into the 6 carbon sugars. Now because there is limited electron transport in these cells and there is no oxygen evolution in these cells, that's why there is no photorespiration present here. The final step